From Microbe TV, this is TWiP, This Week in Parasitism, episode 198, recorded on September 10th, 2021. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. Gorgeous day out there, beautiful. We've discussed this already on TWiP earlier today, so I'll spare the details, but it's one of the best days of the year so far.
Also joining us from New York, Daniel Griffin.
Hello, everyone.
And from Glasgow, Scotland, Christina Naula.
Hello, everybody. It's nice to see you again.
What's the weather like?
It was actually muggy, kind of humid, hot, not very pleasant, not sunny either. Unusual for this time of year.
Very non-Scottish. That's right.
Yes, very non-Scottish. I think we had the hottest September day recorded in a long, long time 2 days ago. And it was absolutely boiling.
Boiling? Wow. I bet it was 25 C that you...
That's right.
I think it was 27.
27, oh.
Yes, very unusual.
All right, it is time to see how our listeners did on our clinical case. Daniel, remind us what this one's about.
All right, for... everyone tuning back in, and for those tuning in for the first time, last TWiP, the case from TWiP 197, we discussed a gentleman in his late 20s, and he was seen by me because he had had several weeks of watery diarrhea. It was not getting better. He shared with me, that he had recently taken a trip to Oaxaca, Mexico. He volunteered, a lot of this history was very fun. He just gave me the history straight out. He told me he had stayed at a nice hotel and he could not understand how he had gotten ill because all the fruits, vegetables, salads had been well cleaned. He was careful to eat all his meals at the hotel. But he did develop diarrhea when he was there for his 2-week stay. This diarrhea has now persisted for several weeks after his return. He went there with 3 other friends. They all stayed at the same place. No one else was sick. He does report that he's otherwise healthy, no past medical history, no prior surgeries, no family history of issues. He does not take any medications on a regular basis, no toxic habits. Otherwise, he looks and feels fine. And as I mentioned last time, I recommended one test, and then we quickly arrived at a diagnosis followed by successful treatment.
All right, we have a good number of cases. Let's start with you, Dixon.
Patty writes, "Hi, Twippers. Patty here in Ireland, where it's 14C, damp and cloudy. Long-time listener, first-time guesser. I will be as brief as the case clues were. Watery diarrhea, salad, and Mexico. I suspect Cyclospora cayetanensis, probably determined using microscopy or nucleic acid amplification testing. For treatment, PD7 suggests trimethoprim sulfamethoxazole or ciprofloxacin. Regards, Patty, a lab tech that loves the Twixt audio gallery."
Christina.
She's muted.
Yeah, I just noticed. Sarah writes, "Dear Drs. Racaniello, Despommier, Griffin, and Naula, I've recently stumbled across your podcast and I'm absolutely enthralled. Here is my first attempt at a diagnosis for your case. I do hope this makes it in time. For this 20-something-year-old HIV-negative man with chronic non-bloody watery diarrhea after having indulged in all kinds of fruits, vegetables and salads while in Mexico under the unfortunate assumption that hotel prepared equates to safe from microscopic creatures, I'm guessing Cyclosporidium, an infection caused by Cyclospora, species endemic to Mexico, commonly found on fresh herbs like cilantro, as well as in salads. I believe it can be easily missed on routine ova and parasite test, but an acid-fast stain could do the trick. Or did he send Cyclospora PCR? Is there a reliable stool antigen test? I wish I knew the exact species, but I'm going to look it up after sending this email. If this person were immune compromised, I would also consider Cryptosporidium, which is Cyclospora's much smaller acid-fast staining friend. Thank you for the delightful experience that is your podcast. You've brought immense joy and learning to my life, and I'm truly grateful. Sarah.
Sarah's an MD student at Penn. There you go, Daniel. We attracted another one.
This is great. Well, Alexander writes, is it my turn, Vincent?
Yes, it is. Go ahead.
Dear TWiP, as the differential diagnosis for diarrhea, acute going on chronic, is very wide, this gentleman is very lucky to have entered into a case for a podcast about parasitism, which narrows my considerations down considerably. My differential diagnosis is as follows. Helminths, while many helminth infections can cause diarrhea, there are usually other symptoms and some probable exposure. It's therefore unlikely to suspect just one of them based on this history alone. Protozoans, a variety of protozoans commonly cause diarrhea. Among them are Cryptosporidium species. Incubation time is about 1 week. Non-bloody watery diarrhea is also typical. 4 weeks is not an unreasonable duration for the symptoms and could even go on much longer in immunocompromised patients. The low inoculum needed for infection would point towards clean-looking food as a possible source for infection. A fecal antigen test is quite sensitive and would produce a quick result. Cyclosporidium cayetanensis. The symptoms and history fit well here, as does the country. Several outbreaks due to contaminated food in the U.S. have been traced to Middle and South America. The route of infection is not so clear, as cysts need to sporulate in the soil before they become infectious, making direct fecal oral infection unlikely. Diagnosis can also be challenging, as several stool samples need be examined. PCR is also not widely available. Cytoisospora belli, or as with Cyclosporidium, route of infection and diagnosis do not align well with the history here. I would also expect the patient to present with more pronounced systemic symptoms. Entamoeba histolytica, I'd expect more systemic symptoms and abdominal pain. Also, the incubation period of 1 to 3 weeks would make it unlikely that he got infected while in Mexico, which is, of course, not necessarily the case. Giardia duodenalis. This is the most common intestinal parasitic disease in the United States. While some patients with giardiasis complain of copious flatulence, foul-smelling stools, and bloating, this is not necessarily the norm. Diagnosis is also possible via antigen test, and infection can occur directly via the fecal-oral route. While the diarrhea in giardiasis rarely lasts 4 weeks, the infection can leave a temporary lactose intolerance in its wake, which could produce similar symptoms. In my opinion, crypto is the most likely diagnosis here, while giardia is also possible. The presentation is not typical, and I wouldn't expect Dr. Griffin to just order a test for giardia based on the history. All the best, Alexander from Vienna, 19C, 66F, and cloudy.
Luke writes, in the spirit of Andrew from Pongaroa, I will say Verte from the always sunny Parntway, also known as Alice Springs, Australia. Wow. Arente is the language of the traditional owners of this region, but the town hosts people from all around the region and the world, while Piri, Anmatiere, and Alyawar people from the north, Pintubi Lutetia to the west, and Pitanjarara to the south, among others, and then a mix of Australians from the past 250-odd years of immigration. I know I butchered every one of those.
No, you were pretty good, actually.
Pichanjatara. That last one, boy, that's pretty hard. Oh,
Yeah.
Whew. Sorry, Luke, and your fellow traditional owners of the region. I listened to the last few episodes of TWiP and a few TWiMs on my drive back from holiday in Darwin. I know Darwin, a fairly corner deficient 15 hours on the road. My guess for your tours to Oaxaca with persistent watery diarrhea is giardiasis caused by Giardia duodenalis. Daniel likely sent stool for microscopy with direct fluorescence treatment with tinidazole 2 grams once or metronidazole 2 grams daily for 3/7. Thanks for all the tweets, Luke. Dixon.
Yes. David writes, Dear Honorable TWiP lass and lads, after skipping an episode because I was too late with the answer, I tried to find out what happened with this otherwise healthy young man. As usually, when there is a little to go by, my mind goes blank. I stare into the distance and use the search function on the PDF of PD, whatever the many advantages of paper may be. A digital search takes under a second. Using watery as the search term, only 7 parasites came up, distinguished or distributed over 19 hits. The first ones to drop off my list were Strongyloides stercoralis. I assume a larva penetrating the skin would have been alluded to. Diphyllobothrium latum. Raw fish consumption would have been an event that should and would have stood out in the patient's mind when identifying suspicious food sources. Then there is Cyclospora chiatinensis. PD7 mentions transmission through raspberries, basil, snow peas, and mesclun lettuce. With a diet consisting mainly of fruits and vegetables, it seems like the obvious culprit and definitely my favorite diagnosis. However, the same revered book also states disappointingly that in immunocompromised patients, symptoms can last up to 2 to 3 weeks. The youngster has had diarrhea for 4 weeks now. Besides that, the clinical picture does not seem fitting. While a delayed onset of symptoms can be accounted for because of the length of his stay, there are no other frequent symptoms like nausea, vomiting, anorexia, or cramps. So bye-bye CC. Cryptosporidium parvum paints a similar picture. While the infection may be everything between asymptomatic and profuse diarrhea, there are other common acute features that are not present. And again, C, in an immunocompetent host is self-limiting. Cyclospora belli is also very unlikely, as that there are many symptoms, and it is rare to manifest itself in immunocompetent hosts. As the patient is HIV negative, it really does not fit this presentation. 2 suspects remain, Entomoeba histolytica and Balantidium coli. The former can become chronic when untreated, and while heme is detectable, it may not be visible to the naked eye. Also, the absence of other symptoms does not really seem unlikely. The latter presents mainly acute symptoms, and while chronic infection has been studied in endemic areas, this seems to suggest that continuous exposure has something to do with it. With a certain confidence, my guess is therefore E. histolytica. As Dr. Griven undoubtedly went through the differential in a flash, I expect he tested for heme and stool and treated with metronidazole. Having won the book a few episodes ago, I remain a faithful and thankful listener in a dreary land, Nicaragua. David. Yeah.
Christina.
Andrew writes, Kia ora from Pongaroa. No book won yet, but I was one away last time. Am I getting closer? Will I win on TWiP 200? By the time this gets read out, I hope New Zealand has returned to having no community cases of COVID-19 and that we are reaching a high level of vaccination coverage. Having an outbreak has motivated many to give up their hesitancy and get the jab. Clouds and silver linings. Speaking of clouds, there are none here and the temperature is cool 14°C. My guess for the gentleman in his 20s who has recently returned from Mexico and has developed persistent diarrhea is Giardia. This is a numbers game as it is the most common cause of diarrhea that persists and is connected with travel to Mexico. I surmised that the test was a PCR and the successful treatment was metronidazole. Na, Andrew.
Wow.
All right. Elise writes, Dear TWiP, quadfecta. I hope this finds you well in this strangest of seasons when the pandemic is still so very present. But there is also a return to school with all of the interest and energy and excitement to rise above it. I am writing with an attempted diagnosis for the gentleman who was visiting Oaxaca. I think he may have experienced a Cyclospora infection, which is unpleasant but not uncommon. There are a few parasites that can trigger the patient's most obvious symptom, watery diarrhea, but I feel that Cyclospora matches best. With a Giardia infection, the loose stools tend to be greasy and foul smelling, and there is accompanying cramps, gas, and nausea that were not among the patient's complaints. Another possibility I considered was Cryptosporidium infection, but I was less inclined toward crypto, a culprit, because in healthy individuals, it seems that the infection is pretty self-limiting, even though the symptoms are very similar to the patient's. In addition to a symptom comparison, I was, of course, sensitive to the discussion of the patient's diet in Mexico and his having eaten a lot of fresh fruit and vegetables and salads. Fresh fruits and vegetables are very common sources for the Cyclospora protozoa, and it is likely that his healthy eating impulses inadvertently got him infected. The diagnosis can be made with an examination of a stool sample, and treatment is with trimethoprim, sulfamethoxazole. Some brand names for this are Bactrim and Septra. I do have some questions. Is it just luck that his travel companions didn't contract the infection if they ate the same food? Is it more likely that they consumed only cooked produce? Is there any way he could have enjoyed fresh fruit and vegetables without risking an infection? I've heard that if fruit is washed and then peeled, the risk could be reduced. As always, thank you for your work and your energies. Best wishes, Elise in Lower Manhattan.
So Elise has the ER in big caps. Is she suggesting that it's a big ER in Lower Manhattan, Daniel?
I was not sure what that was. Lower Manhattan.
Huh. All right. Martha writes, Dear TWiP team, sussing out the parasite in episode 197 felt like solving a good old whodunit. A young man and his companions had traveled to Mexico. They stayed in a nice hotel where they dined almost exclusively. He could tell it was a good hotel because of all the fresh fruit and vegetables being served. He's now plagued with 4 weeks of watery diarrhea. The infectious disease physician listens to his story, orders one test, and on getting the test result, prescribes a medication. My thought process was to find the parasites that cause prolonged traveler's diarrhea in an immunocompetent host. Check for those that have a treatment other than supportive care. Cross-check with those that have a unique diagnostic test. I ignore viral and bacterial causes of diarrhea because the podcast is This Week in Parasitism. It's part of the differential, right? I assume that Dr. Griffin had other reasons for eliminating the virus and the bacteria. Giardia and entamoeba both can be diagnosed by ELISA and can be treated by metronidazole, but neither is described as the cause of watery diarrhea. Cryptosporidium is diagnosed by ELISA, but is reportedly treated by supportive care. Microsporidia is allegedly self-limited in the immunocompetent, which brings me to Cyclospora, which, although usually self-limited, can cause prolonged watery stool. Cyclospora autofluoresce under UV light at wavelength 300 to 380 nanometers. Based on this alone, I want Cyclospora to be the answer because being able to glow blue is just too amazing. Treatment is with trimethoprim, sulfamethoxazole. Cyclospora outbreaks are linked to a variety of fresh produce. So in the fashion of Clue, it was Cyclospora in the dining room with the salad bar. Best wishes to you all, Martha.
Cool.
Dixon.
Katie Jane writes, Hi, everyone. A pleasant 62F/17C this morning in north central Wisconsin. I like to consider myself a fairly well-traveled person, and in this case struck a chord with me. I shan't divulge further. There are many causes of traveler's diarrhea, most of which usually resolve fairly quickly on their own. Common culprits are our friends, E. coli and Clostridium species, but according to the CDC, bacterial infections rarely cause persistent infections. Also, this is a podcast about parasites, not bacteria. Persistent diarrhea occurs in a minority of cases and is usually caused by protozoa. Giardia species, Cryptosporidium species, Cyclospora species, and Entamoeba histolyticum. protozoa like wet environments, so it is highly likely that our patient contracted his disease from eating nicely washed fresh fruit in the hotel. Giardia causes watery diarrhea and is the most common protozoan cause of traveler's diarrhea, so this is what I'm going with. I found the following quote on a Banner Health website, which I thought was good advice. Cook it, boil it, peel it, and when in doubt, leave it. Thanks always, Katie Jane.
You can't peel lettuce, can you, Dixon?
No, not really. You can shuck it, but you can't peel it.
No. Christina.
Cheryl writes, hello again, Vincent, Dixon, Daniel, and Christina. The case presented is of prolonged watery diarrhea in a young, healthy male traveler with no relevant medical history. Recently returned from
Oaxaca.
Oaxaca, thank you, Mexico. Relevant clue given is that patient ate lots of fresh fruits, vegetable salads, mostly at his nice hotel. I find this case quite tricky to pin down because there are so many infections that include watery traveler's diarrhea. Such diseases are abundant in Mexico and are sometimes referred to as Montezuma's revenge. Here's my working list, by no means exhaustive. 1. Entamoeba histolytica. This is a worldwide protozoan transmitted by fecal matter in water or soil. Cysts can lie dormant in the environment, contaminating fresh water and food. Norovirus, common stomach virus transmitted in uncooked raw food, water, and on contaminated surfaces. 3. Cryptosporidia, another protozoan involving fecal contamination of water or uncooked food, resistant to chlorination and thus can be acquired from pools. 4. Giardia, one more intestinal protozoan parasite, flagellated, that is transmitted via fecal matter in fresh water or uncooked food. 5. E. coli, rod-shaped bacteria transmitted through water, uncooked food or soil. I better shorten up these notes or this email will be too long. 6. Shigella bacteria. 7. Salmonella bacteria. 8. Campylobacter bacteria. 9. Vibrio bacteria. 10. Rotavirus. 11. Enterovirus. 12. Cyclospora cayetanensis. My best guess is Cyclospora cayetanensis because my Google research shows this one most closely fits the fact. Weeks of watery diarrhea and no other symptoms reported after eating a lot of fresh produce in Mexico. The Cyclospora parasite is coccidian and oocytes are ingested on fresh produce. The other contender was Giardia, which might be more common, but the clue here of persistent diarrhea seemed to fit Cyclospora better. Giardia symptoms will probably include foul-smelling gas and bloating. And Giardia seems to be acquired more commonly from contaminated fresh water than, say, salads. The treatment for Cyclospora cayetanensis is oral TMX, SMX, Acabactrim. Same drug I have given my horses to treat infections many times. I learned so much from this quest, but admit I'm still struggling to master the correct spelling of diarrhea. I have the same problem. I take it that H is silent. Many thanks. Here's a link I find convincing on this one, and he provides a link by medicinenet.com. Cheryl. Apologies, Cheryl.
So, Christina, you pronounce it H, right?
I do, yes.
Yeah, but you picked that up in the UK because you're from Switzerland, right?
Yes, yeah, I'm German speaking natively. So.
Yeah, because we say H. It's interesting because I always hear Brits saying H, and I have on my video equipment an HDMI port, and they all say HDMI. It's really strange. Not strange, just the way they do it, but you picked it up. All right, sorry, Daniel.
That's okay.
All right. Well, I think Christina can tell me her friends asked, her children asked at one point about me, Mommy, why does he speak so funny? I was speaking, the American version of our language there in Scotland. But all right, the University of Central Lancashire Parasitology Club writes, Dear TWiP professors, hello again from the Parasitology Club at the University of Central Lancashire in the beautiful northwest of England. We recently enjoyed our first parasitology summer school. With socially distanced microscopes, we studied a variety of enteric protozoa and helminths. We played at dung from horses to discover which had been recently wormed, and played a game called Which Parasite Would You Most Rather Avoid? Our collective investigations in the case of the young man with persistent watery diarrhea who recently visited Mayco led us to a couple of coccidian candidates in Cryptosporidium or Cyclospora. Cryptosporidium is a cause of acute diarrhea, which normally resolves without treatment within a week or so in the immunocompetent individual. This can be transmitted via contaminated produce and water. We considered the possibility that the subject may have ingested oocysts while swimming in the hotel pool after a fecal accident, as we know that crypto is highly resistant to chlorination, and there have been outbreaks associated with swimming pool water. However, because of the prolonged episode of diarrhea, we believe that this is a case of cyclosporiasis caused by the parasite Cyclospora cayetanensis. Cyclospora is also highly resistant to chlorine disinfection. Therefore, it could be transmitted by ingesting contaminated produce or water, even when vegetables and fruits are washed in potable water. Outbreaks of cyclosporiasis have been reported worldwide associated with soft fruits and fresh produce, including a few outbreaks associated with coriander leaves, cilantro, from Mexico. When most healthy individuals become infected, symptoms may persist for several weeks without treatment. Stool examination by either differential interference contrast, DIC microscopy, or fluorescent microscopy at 330 to 360 nanometers should reveal 8 to 10 micron round oocysts with a distinct oocyst wall and internal sporozoite structures, or a beautiful blue autofluorescence, respectively. Trimethoprim sulfamethoxazole is the usual therapy for Cyclospora infection. Thank you, as always, for your wonderful podcast and stimulating case studies. We are keeping our fingers crossed for a signed copy of your wonderful book to make the long voyage across the Atlantic to Lancashire. What do you call a magical poop? Poop Dini. David, on behalf of University of Central Lancashire Parasitology Club.
Houdini.
Poodini. How did I pronounce that?
Poop. You said Poopdini. You added a P.
Okay, Poodini. I added the extra P there.
Yeah, well, maybe we're getting towards the day where we could start signing books.
I think very
Daniel,
soon,
I think the pronunciation of their school is Lancashire.
Lancashire. How was I doing that?
Lancashire. Lancashire. They sort of smooth through the last part. Lancashire.
All right, that's it. We got a nice number this time, and 8 of those are eligible for a book draw. But
right,
let's see how this sorted out, Daniel. All right. Well, I think we have 3 more guesses.
We do
left. So let's start with you, Dixon.
Sure, I knew you'd do that.
I'm calling on you. This is kind of like a turnaround, right? Because
that's right.
I'm calling on my professor.
That's okay. I think I can get this one right. Okay. I'm going to go with Cyclospora cayetanensis because of the symptoms. Basically, it's just a simple watery diarrhea that lasts for a protracted period of time. I think that you could probably distinguish that between Cyclospora and Cryptosporidium, but it certainly is not anything like Giardia, because Giardia produces foul-smelling stools and a lot of flatulence and things of this sort. So I wouldn't count Giardia in. I think Cyclospora is the logical choice here, and probably in some fruit cocktail that he received at the end of a dinner, maybe with some fresh raspberries on top, and that would have done it right there for me. And you would never suspect that in a food source, but it's very common, unfortunately. So that's my guess.
All right, Christina, are you ready?
Yes, I am actually. So I had good time to think about it. So I think it's really 3 contenders. I would say it's a protozoan parasite, not a worm.
Right.
And so we have Giardia and Cryptosporidium and Cyclospora. And I think, as Dixon said, I think Giardia is kind of maybe initially watery, but I think sometimes turns into fatty diarrhea. So I would really exclude that. And then also the diagnosis, you'd probably need to do multiple stool samples. So I don't know if that's summarized as one test, Daniel. If it was done multiple times, I don't know. So, and then cryptosporidiosis, I think I'm going to exclude that because you treated the patient, whereas I think there isn't usually treatment for Cryptosporidium unless it's a HIV patient, and you would treat them with antiretroviral therapy. So I think I'm also going to go with Cyclospora cayetensis. And I presume the test was microscopy, but I don't know what fancy tests you might have available, Daniel.
We've got fancy tests here in the US.
Yeah, so maybe have a PCR or some ELISA, but I'd probably settle on microscopy, where you said see the small little round spheres. Quite pretty, actually. So my guess is on Cyclospora cayetensis as well.
All right, Vincent.
This was difficult for me. I wanted to pick E. coli, but I know this is TWiP, and I think that's an artificial exclusion that we have. But I guess we assume that Daniel is treating parasitic infection, so we can always rule out, or at least we won't hear about those. So I had a trouble between Cryptosporidium and Cyclospora, but then I remembered asking about the HIV status of this patient, HIV negative. So that and this nice diagnosis with the fluorescence made me migrate to Cyclospora.
Okay. Yeah, hopefully I was giving people that little extra clue by saying, and then I treated it, right, to help with that distinction. There also was the duration, right? So this had been going on for weeks. It was not self-resolving in this young, healthy individual. So I do think that when you initially hear the story, this watery diarrhea, Cryptosporidium, Cyclospora are kind of in the differential. It's not a bloody diarrhea. There's no fever. There's no foul smelling. There's no flatulence, abdominal discomfort. This is kind of a classic presentation, I think, of Cyclospora, or I thought at that point. And what I sent off was we have the ability to send off these GI PCR paddles. And I don't know if our listeners are familiar, but they're really, it's really a symptom approach to diagnosis. You don't really even have to know ahead of time what you're thinking about. Cryptosporidium, Cyclospora, Entamoeba, Giardia are all in there. There also are viruses, right? I wasn't thinking, norovirus, usually there's a vomiting component. It doesn't have the same duration. Rotavirus, sapovirus, astrovirus. There also are the bacteria, and I think the E. coli that Dixon brings up, that's actually the most common cause of traveler's diarrhea, but usually it's not going to last this long. I guess maybe an enteroaggregative E. coli I've seen last a little bit longer. But again, all those targets are in there. But also Campylobacter, Clostridium, Salmonella, Yersinia, the Vibrio, those are all in there. But really what I think pushed me to thinking ahead of time was the duration, the fact that this individual felt well otherwise, was just this watery diarrhea that was persisting. So yes, the test came back positive for Cyclospora cayetensis. Actually came back like later that same day that the stool was collected, put the gentleman on the sulfamethoxazole trimethoprim. That's your Bactrim or your sulfa. And he actually got, or Bactrim or Septra, and he actually got better really about, I'll say 36 hours later. So really like very quickly started to feel better, completed therapy. And that was the end of it.
Was that your guess also?
That was my guess. That was my guess ahead of time.
Okay, good. Now, why didn't his friends get it?
Yeah, I don't know. I mean, I think that this gentleman may have been just consuming so much fruits and vegetables. And
the raspberry lover.
So, yeah, I suspect that was what happened there.
So, Daniel, how do you avoid this? What do? You go to a nice hotel in a certain country. What do? You stay away from the fruits and vegetables?
There's three things that, well, I'm going to say maybe it's four things that human beings do that get them in trouble. One is we breathe, 2 is we like to eat, and 3 is we like to drink. And so every time you do one of those activities, you're at risk. The human condition is always at risk. So there is that advice, right? That we heard, peel it, wash it. The interesting thing is that when you give people that advice, they've done these prospective studies. The people following advice are just as likely to get sick as the people not following it. So that doesn't make a lot of sense to me. I must admit, like maybe I follow in the Mark Crislip approach when I travel overseas. I like to eat things that are deep fried, that are cooked, that are hot. I'm not a big fan of the salads that are sort of put out there, and it isn't always, the fancy restaurant that I feel necessarily safer than I feel at some of the other places. I like food that's cooked, food that's hot when I eat it. But this is one of the risks that we take when we travel, particularly to more exotic locales.
Oh, Mexico's not so exotic, right? But
yeah, this is always my excuse for drinking soda and beer instead of, other options.
Water. All right. It's time to give away a book. We have 8 eligible people. So let's do a random number between 1 and 8. Well, number 8. Number 8 came up that time. That is, the University of Central Lancashire.
There you go.
And
you've got a team. You've got a team now. I don't know if you can just give it to one person.
Well, they're going to give me an address, and it's going to go to the address. They'll probably stick it in the library somewhere.
Good deal.
So, David, send us your address, TWiP@microbe.tv. Also a phone because it's overseas. And sometime in the next millennium, you'll get a book autographed. We'll get together and autograph. We have a whole pile, as you can see behind me here, of books, but these are not autographed.
Right.
So at some point, I will bring them to the incubator, our new studio downtown, and then as people visit, they can autograph them.
Right.
So please send us your address, and the other ones of you keep trying, because as people win, they're pulled out of the pool. So you have a higher and higher chance of winning.
That's true.
Well, thank you all for your guesses. All right, on to a paper. Now, for the second episode in a row, a paper has been selected by Christina. So she has the honor of presenting it to us.
I'd be delighted to do that. I'll do my best.
Christina, is it true that your husband picked this paper?
It is indeed true. I was just about to say that. We talk a lot about work and parasites, and he sent me an email with a whole raft of papers that I might find interesting, and that one...
is he a parasitologist as well?
He is a parasitologist, so he's a Leishmania. So I think that's his favorite parasite. He works with Leishmania. So, yes, but didn't choose a Leishmania paper. Proposed this malaria paper. And I think in the future, I need to pay attention, because this was a paper that is very complicated and has lots and lots of experimental details. And I kind of got myself down a rabbit hole while I was reading it. So I think I'll summarize it, but let me first give you the title. So the title is a heat shock response regulated by the PFAP2HS transcription factor protects human malaria parasites from febrile temperatures. The first author is Elizabeth Tinta-Font, and the corresponding author is Alfred Cortez, with quite a few authors in between, as you would expect, I think, from a Nature microbiology paper. So I hope it is acceptable if I kind of give a summary rather than a detailed insight into the experimental procedures, because I think that might take several hours.
I was going to encourage that.
Good.
Yeah, this paper is very complex, but I think the gist of it is important, very interesting.
Yeah. And actually, so really the main point is that malaria causes a high fever, so around 40 centigrades, or I think that's maybe 103, something around 100. a little bit more than 100 Fahrenheit. I think, please correct me, I'm not familiar with Fahrenheit all that much. And so this paper really looks at how the malaria parasites withstand the fever. So how can they survive those high temperature? Because most eukaryotic cells are quite sensitive to heat, stress. So this is really the kind of, the main bulk of the paper looks at that. So I don't know if I should maybe explain the basics of heat shock response, or would that be too much? I don't know. What do you think?
Yeah, I think if you can summarize
just the very basics.
Yeah.
If I can't find my notes, but I've got some notes somewhere. Here we are. Got so many notes. So all our cells they often have stress and from their environment. So that could be heat, or that could be chemical stress. For example, when there is drug treatment, and so the stress that can cause protein damage. For example, the protein in the cells, they maybe stick together, so they aggregate, or maybe they lose their correct three-dimensional shape, and that really stops the proteins from being functional, which is problematic, of course, if they have an enzymatic function, for example, but also problematic if they maybe have structural functions. So the heat shock response is really a program that acts at the level of transcription. So when genes are transcribed into RNA, and this is a response that protects cells from heat-related stress, but actually also other stressors. And this is conserved through evolution. So most eukaryotic cells, or probably all eukaryotic cells, maybe some weird ones like, I don't know, some weird protozoan eukaryotes have different heat shock responses, but this is really very well conserved. And the response is orchestrated by one transcription factor, which is the same in all eukaryotic cells. And what a transcription factor does, it binds to DNA, and specifically to the regions in the DNA that regulate how much of a gene is transcribed into RNA, so how much protein is ultimately produced. And this transcription factor binds to specific genes that code for proteins that are called chaperones. And I think in the name you already have, explained what they do. They essentially
keep you from having fun? Is that?
Yeah, keep you from having fun, I guess, yes.
That's what I think of when I hear chaperones.
Yes, I was kind of taking a more positive spin in that they are protective, they're looking out for something, and they actually can help misfold the proteins, or they can direct them towards degradation. So when the proteins really can't be fixed, they are going to be labeled, this is rubbish, you need to get rid of that. And then, so when there is a lot of stress, these chaperones are produced a lot more frequently and in quite high amounts. And then once the stress has been dealt with, these chaperones just return to their normal levels. So that's the heat shock response in a nutshell, really. Now, in Plasmodium, that is a bit unclear. So proteins like, for example, chaperones, they have been found, but it wasn't really clear how they were regulated on a transcriptional level. So we have all the protection mechanisms there, but it just wasn't clear how they were switched on. And this is what the paper really describes. So, and it is all these experiments are based on a cell line that was present in the lab that kind of showed mixed responses to heat shock. So when they were heating that cell line up to 41.5 degrees, I think it was. So some of the cells that kind of dealt with that very badly, but others did okay. So they assumed that must be a mixed population, and they were cloning these cell lines out, and they ended up with 2 cell lines that were sensitive to heat shock, and they called them R1 and R2. What they then did, they kind of analyzed the transcriptome of these cell lines and compared them to a cell line that was resistant to heat shock, so it could handle the heat. Now, the transcriptome, that's the combined every single transcript in a cell. So the whole pile of RNAs essentially. So they were comparing that. And they found one single point mutation that caused the sensitivity to heat shock. So it was really only one single base change in one protein that made those cell lines sensitive to heat. And that's called a SNP, a single nucleotide polymorphism. And what that SNP does to a protein that we have yet to identify is it truncates it, so it inserts a stop codon and the protein ends up being shorter than the real thing. And they identified the protein as PFAP2HS. I think I'm just going to call it the transcription factor for simplicity. It just kind of rolls off the tongue a bit easier. But if you want me to say the whole thing, I can do that too.
I'd like you to say it 10 times each time.
All right, let me get started. PFAP2HS. So they had these SNP mutants with a shortened protein, and then they thought, well, how about we go on to just delete that entire gene that codes for that transcription factor? We'll just get rid of that altogether. And they did create knockout mutants using CRISPR-Cas. And they did that in 3 different genetic backgrounds, I suppose, just to be, on the safe side, or just to be, just to cover all what a potential. Sometimes if you only look at one cell line, you kind of think you see something interesting, and then when you eventually finally look at other ones, you realize, well, actually it was just an artifact. So I presume that's why they were looking at 3 different backgrounds, but I'm speculating, so could be different. So they did actually find it quite difficult to make those knockouts, so they had to decrease the culture temperature of the malaria parasites so that they could actually even create those mutants. But they managed to get these knockout cell lines, and they were very, very sensitive to heat shock, much more so than the truncated, the cell line with a truncated version of this transcription factor. So then they did lots of experiments comparing the transcriptome of these normal cell lines, but also the cell lines with the truncated protein and the ones that had no transcription factor, PFAP2HS at all. And it's really complicated, but I think the take-home message is that the heat shock response really turned out to be regulated. The heat shock response that was regulated by this transcription factor leads to a very quick upregulation of 2 proteins that happen to be chaperones. So I think that kind of follows on quite nicely. And these 2 chaperones, they're a bit differently, so upregulated to different amounts. So one of them, HB71, is I think maybe 16-fold upregulated and really, really quickly. And the other one, HSP 90. Yeah, is upregulated maybe 4-fold, but quickly and very considerably. So then they wanted to find out, how are these chaperones upregulated so quickly? And they found out that the transcription factor, PFAP2HS, binds to a twice repeated stretch of DNA. I think that is called G-box. And that stretch of DNA, those 2 little elements, they are located in the parts of the DNA of the gene that is not transcribed into RNA, so the regulatory sequences So I think that's quite nice. So, the transcription factor binds to that. And actually only that, they found very little other proteins that would also bind that transcription factor. One of them was a small nuclear RNA that they didn't follow up. And another one, I think I can't remember the other protein, but there was a third one. So apologies, I can't remember why they didn't follow that through. Oh, I think they did, but I just can't remember why it's not becoming relevant. So,
our listeners always ask us for an executive summary. So that was really nice. And now Dixon will go, I guess, into depth for us. I'm joking.
I'm not quite finished, but I'll just have a few more sentences.
If I went into depth, Daniel, I would have been diving off a board into a very deep pool.
No, this is excellent so far, Christina. Please continue.
So I should probably mention that if you remember back, there was a truncated version of that transcription factor, and that truncated version actually misses one of the 3 DNA binding domains that this protein has. So, and it turned out that this domain in particular, and only this, is essential for the upregulation of the chaperones. So if that is missing, then the chaperones are not upregulated, and therefore the malaria cells can't withstand the fever, so they would die. As a side, they've also found that actually the heat shock leads to a delayed growth of the protein, the parasites, but that's probably a bit less relevant. And they also found a whole raft of other heat shock response genes that are upregulated, but in an independent fashion, so not dependent on that particular transcription factor. And these independent responses, they are not enough for the survival of the heat shock. So they probably help with the kind of follow-up repairs and whatnot is needed, but they are not, they're not enough on their own to make it possible for the parasite to survive. So this really means that this PFAP2HS transcription factor is essential. I forgot something got really, really relevant. So actually they were looking at a lot of different cell lines, and they found that truncations of this protein, they are often found in culture, but they are hardly ever found in clinical isolates. And they looked at a lot. They said they found only 1 in 2,500 isolates that they looked up did they find a truncated version. So I find that on its own, I find that astonishing. I mean, how do you look at 2,500 different isolates? Mind-boggling, really. So this is really, it just means that this protein, we can conclude from that this protein is essential during natural malaria infection, but not really needed in culture. And that makes sense because in culture, that there is no fever. You culture those parasites at the comfortable 37 degrees. So that kind of would explain why you find the truncated version maybe in cultured isolates, but not in clinical ones.
I was suspecting there was some kind of a cost to maintaining this, some sort of fitness cost to maintaining it.
Yeah.
So what it doesn't need to if it's not under pressure. Actually.
Yeah. Actually, in a very final experiment, and I thought that was really nice, that actually also showed that these cell lines are also more sensitive to artemisinin, which is really the main drug treatment used for Plasmodium falciparum and malaria. And artemisinin really also induces stress that can affect the proteins. So that kind of makes sense. So I thought that was a kind of a final nice experiment to show that. So that I think that's the paper in a nutshell. There's a lot more to it, but I don't think it's an open access. So I think it would make little sense looking at individual figures and explaining those in any detail.
So you say the What is the temperature that malaria induces in people? Is it over 39 or is it 39? What is it?
Yeah, actually, it uses a really high fever, doesn't it?
Yeah, so it's 40 C, which is about 104 Fahrenheit.
40
these are
C.
these are very high fevers, which is really interesting, right? Because we normally think, right, like as physicians, whether or not we care about fevers or not, the nurses will make sure we do at 2 a.m. And so, I like to think that fever is doing something helpful. So, a fever of 101, a fever of 102, but these are 104. This is the fever so high that person is delirious. And so it's interesting, right, that the parasite is somehow able to withstand that. And I don't know if people know the sort of the Nobel Prize story about infecting people with malaria as a syphilis treatment. Dixon's got to be someone who knows about that.
I was just going to say that, Daniel. I've got a note of that as well.
Okay.
It's really amazing, isn't it? So you carry on, though.
Yeah, when you get up to that high temperature, that actually most organisms, the treponemes have very primitive heat shock protein chaperone systems, so they can't handle that abrupt rise.
So, Daniel, is that also true for viral infections, or the fever is not generally as high?
It tends to be not as high with viral infections.
So, fever is not playing as big a role in clearing viral infections then?
So I don't know. That's an interesting issue. We tend to think of it as important, but how good is the data supporting that contention? It's not great.
So how do the eukaryotic cells of the host fare in these high temperature situations? I guess their heat shock proteins deal with it, right, Christina?
I think they must do. Actually, the authors didn't mention that, but I just assumed that would be the traditional kind of the same heat shock response, but driven by a different transcription factor. I think it's called HSF1 in all other eukaryotic cells. And actually, the malaria protein is not similar to that at all in its amino acid sequence, but it seems to have an analogous function. So that's quite interesting as well, that malaria just have have a protein that does the same thing, but it's really quite different. Yeah, I just wanted to go back to that syphilis treatment, Daniel. I thought that was so amazing.
Let's you.
Yeah. No, I just thought, they thought, obviously that was before there was antibiotics to treat syphilis. And I just thought this is just so astonishing that they would treat patients. They would essentially infect people with vivax malaria, so they used Plasmodium vivax, which is maybe a bit less severe than Plasmodium falciparum.
Correct.
And so they use that because that would treat, or that would essentially get rid of the fever induced by the malaria, would essentially get rid of the syphilis. And then they would just treat with quinine, I think. And that was that. So they had effective antimalarials, but no effective antibacterials. And I thought it was really interesting. So Richard, who reminded me of that story. So I can't take any credit today. It's all my husband.
It's interesting. You're sort of mixing an old world and a new world pathogen together, right? Because syphilis was what the Americans gave to the Europeans, who gave them so many other things, and so many things brought over from Africa. So it's interesting to see, malaria treating syphilis, but we do not do that, nor do we recommend that anymore.
Actually, I was told that Lyme disease.
Antibacterials now. Sorry.
No, there was a recent article, I thought, of treating Lyme disease with Plasmodium falciparum or Vivax rather. I'll try to find it. I heard about that because it's also it's not neurotropic, but the immune system sort of forces it into immune silent tissues like brain tissue, that sort of thing. And it's a long-term.
Not sure if I would consent to that treatment.
And it's a spirochete. What's that?
I'm not sure if I would consent to that treatment myself.
If you had Lyme disease, you might. Because you get so miserable after a while that because there's a tremendous constellation of symptoms. that it drives you nuts, that you just don't want it anymore. So you'd rather take that. And okay, then we'll cure you of that, and you'll be all better. Ha ha ha. I think they used Plasmodium vivax because at that point there was no drug resistance like we have today. I think there is some drug resistance to vivax as well. So not as severe as falciparum, but for sure that it's still there.
Daniel, they suggest that you could target this heat shock protein as a therapeutic. Does that make sense?
It does actually, because it's different, right? It's different than the heat shock protein system that humans, eukaryotic cells have. Yeah. And I think they even sort of brought up the idea that you don't even have to completely shut it down. It may actually increase the ability of some of our other medicines to work. So maybe it gets added in as an adjunct, helping with the resistance that we're running into. So and it is now you understand this whole new system. It's a whole new area to potentially target. So this is really interesting basic science. Really interesting, like what's going on? Why don't these high fevers do something helpful with malaria? But also there is that angle of potential therapeutics.
So the idea would be you inhibit the heat shock protein and then high fever will clear the parasite, right?
Right.
Yeah. Yeah.
So it will knock itself out, really.
Right.
Actually, this protein is found in all Plasmodium species that they looked at. So I think that could maybe be quite a generic treatment. It's even found in Plasmodium species that don't induce fever. I think some of the mouse malarias don't really give a fever spike, and it's even found in those. So it could potentially be quite far-reaching as a potential drug target.
So Daniel, the fever goes up and down, right? So would that be an issue with inhibiting this? Because if the fever drops, then there's no selection against the parasite there, right?
Because really this whole system is only important while a person is having fever is actually sort of the idea here. Yeah. So if the person, but those fevers are going to come, and we normally think of these rhythms, right? And the initial description of the fever coming, every other day, every third day, et cetera. But in a traveler, for instance, they may get more frequent fevers because it takes a period of time before the schizonts and the erythrocytic lysis gets synced. Yeah. So these are our highest risk, highest mortality individuals. If you've had a while for them to synchronize, you're probably going to survive, in all honesty. So the highest risk people are people newly infected before this synchronization occurs. So I don't think we're going to have to worry about there not being enough fever in these sick people.
All right. Well, thank you, Christina.
That was excellent. And boy, did you pick a complicated paper.
Thank you. I know. I think next time I need to make sure I choose one with few experiments. But it was quite nice to get bogged down into an interesting paper. So that's a nice way to spend an afternoon, really.
Yeah, there's the nice, I think you also shared the news and views synopsis that Bandana, that I don't know how to pronounce this. Do you know how to pronounce Thathy or Tati? And David Fiddick. And David actually is at Columbia. He's going to be doing our morning infectious disease conference next Thursday morning. So 6 days from now. So.
Nice.
So you can mention that we discussed. Well, I suppose he didn't do the paper. He just summarized it nicely.
So, yeah. He's got some interesting stuff going on. So it'd be interesting to hear. It's nice to see, malaria. I feel like we've lost some ground in the last 2 years with our malaria efforts. So it'll be interesting to hear where the basic science is because, it is a really critical issue in so many parts of the world.
Dixon, you have a hero for us?
This is happens to be a person who's still alive and well and living in Los Angeles and works at the University of California at Los Angeles. Her name is Claire Panosian Donovan. That's her married name. but I knew her best as Claire Panosian when she was a physician at Tufts University up in Boston. We first met while I was going up to participate in a course that Jerry Kirsch ran and invited me to give several lectures in. And I met her along the way, and she was a very engaging person, very vivacious, very knowledgeable, very interested in infectious diseases, particularly parasitic infections. And she developed a career in parasitology as the result of that. And in fact, she has her own website, so if you want to go look her up, she says, "Welcome to my site, looking for Dr. Claire Panosian, the tropical disease, global health, and infectious disease specialist." That's me. So, you can find out all about her there, and I'll just read the credits that she lists as part of her credentials. She was the 2008 president of the American Society for Tropical Medicine and Hygiene. She has an MD degree plus a doctor of tropical medicine and hygiene from London School of Tropical Medicine and Hygiene. She received her education at Stanford University and Northwestern Medical School, and finally did her residency, and then finally had her first appointment at Tufts New England Medical Center in Boston. She was her first appointment was as chief of infectious diseases in the Los Angeles County / Olive View Medical Center, and then as the director of travel and tropical medicine at UCLA. She is currently a clinician in residence, a UCLA professor, and she's taught there since 1984. She's also worked overseas in Haiti, Taiwan, the Philippines, Pakistan, Vietnam, Albania, Armenia, and Tanzania, among other countries, as she lists. And then finally, Vincent, the reason why I was really impressed with her was her ability to communicate. So she is a science communicator in infectious diseases. She married a gentleman who produces documentary films, and he's an 8-time Emmy award-winning documentary filmmaker. His name is Patrick Donovan, and she produced a show on television on hepatitis B, which has received 300 million international views. In recent years, she has written regularly for national newspapers and magazines. She currently writes a weekly column called The Infection Files, which runs in California newspapers. Her journalism spans issues in infectious diseases and public health, affecting everyone on the planet, to global health policies and economics. So that's my hero for today, and I'm happy to be delivering that information to everybody who's listening.
All right, now it's time for a new case, and today it's not going to be from Daniel, right?
Aha.
Very exciting.
Yes, it's from me, and I'm feeling a bit self-conscious now because I've also got that microphone. But I have no beard, so maybe it's okay.
You're fine. There's no noise
Not
from
yet,
you.
Anyway. Okay, so as you probably know, and as our listeners probably know by now as well, I'm not a clinician, so I don't see my own cases, but I do have a very large group of students who study tropical medicine and hygiene with me. And this particular case today is from one of our tropical medicine and hygiene students, Eyal, and he kindly agreed for me to share it with the TWiP listeners and the group here. So this is actually a really interesting case, and it's quite a protracted case. I posed some diagnostic challenges for the treating doctors. And I will give you full credits next week, just not now, for reasons that will become apparent later. So this is the case of a 30 Dr. Google. This is the case of a 31-year-old woman, and she was previously healthy, but she had traveled to Tanzania, and she went on a quite long safari, I think maybe an 11-day long safari or something like that. So she spent quite a lot of time in the savannah. So 1 week after the safari, which was a day after she returned home, she developed a fever and a really severe headache. And she was actually so unwell that she was admitted to hospital. Her blood cells were not hugely unusual, so she had slightly reduced white blood cells and slightly reduced platelets. And her liver enzymes were were just a little bit increased. So I did thick blood smears at a hospital, and they were negative for malaria and other blood pathogens. And other evaluations were normal, and her physical examination was also unremarkable, although she did have a small purplish, painless lesion near her buttocks. And so the treating doctor thought that this might be a tick Eschar, and he started her on treatment for African tick bite fever, which is a bacterian pathogen. So she was started on treatment, but she didn't really get any better, and the lesion on her buttocks also grew in size. So they did rapid antigen tests for malaria, which was positive, but it was thought it had to be false negative because the thick and thin blood smears that were repeated were still negative for malaria, and they were also negative for other pathogens. And they did a biopsy of the lesion from her buttocks and analyzed it microscopically, and that showed nothing remarkable as well. So after nearly a week, she improved and she was sent home from hospital. But then 10 days later, she had another bout of fever, which resolved spontaneously after 2 days. And in the meantime, her lesion had started healing. But now she was slightly anemic and she had a very high C-reactive protein. And blood cultures and PCR diagnostic tests, which were now ordered for Plasmodium and also Borrelia species, were negative. But she had now seroconverted for rickettsiosis, so it gets really complicated now. And blood smears were taken when she didn't have a fever, and they were negative. So she went home again, and a week later she had yet another 3-day bout of fever. And this is where I leave you now to think about the case. So what could this be, based on the quite long case history that I have given you now? But I think it was really important that you had all that information. So yeah, if you have any questions that are not giving the diagnosis away, I will answer them if I can.
Is she HIV negative?
So I don't have that information.
So let me ask. So this lesion, did she know when she acquired it? Was it something where there was pain, or just, oh, by the way, this was discovered incidentally?
She did remember where she had acquired it after it was pointed out to her in the hospital. So when her doctor asked her, how did you get that lesion? But that's all I'm going to say, Daniel.
Yeah.
Okay. All right. I'm good.
I thought you might be when you asked that question.
Yeah.
All right. Guess the lesion's important then.
Potentially.
No, it's...
Yeah, I could just be throwing a red herring, but yeah.
I don't think so.
I had that for dinner last night.
Herring?
Red herring.
Red herring with onions and sour cream.
That's exactly.
So was the lady alone on this safari, Christina?
I also don't have that information. I presume she must have traveled with travel group, but there was no report of a partner or family or anyone from that travel group becoming sick.
Okay.
So not something that spreads like COVID then.
Okay. And she didn't eat unwashed fruits and vegetables and lettuce, right?
Oh.
Nothing but
She
beer.
might have, but
That's right.
we're not sure about the relevance.
But no, it wasn't part of the report, right? You don't have that information.
No, I don't have that information.
And
Okay.
and I get you described this as a single small lesion. So there was not a group of lesions. It was just one lesion.
Yeah, it was
Okay.
just one lesion, yeah, and on her buttocks.
Okay.
She must have been wearing shorts, I suppose.
Yeah, it was going to be like where on the buttocks, but yeah. So it was we're thinking it was below what was covered by clothing. Okay.
Actually, the description is near her So I need to clarify that. Not so. Yeah.
Near the buttocks. Okay. Yeah, near. That's what you wrote.
Yeah.
That's what you said here. Yeah. Okay.
Yeah.
All right. Anything else, gentlemen?
Oh, this is great.
All right. So
Lots
next,
of unanswered questions.
take your chances, readers, listeners, and send your guesses in.
Exactly.
And that'll do it for TWiP 198. You can find the show notes at microbe.tv/TWiP. Send us your questions, comments, case guesses, TWiP at microbe.tv. We're going to record in about a month on a Friday probably, so you have some time, but don't delay more than a month-ish. And if you enjoy what we do, consider supporting us. Microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, my pleasure.
Dixon Despommier is at trichinella.org, thelivingriver.org. Thanks, Dixon.
Welcome, Vincent. Good time.
And Christina Naula is in Glasgow, Scotland. Where are you, at the University of Glasgow?
Yes, University of Glasgow. Yeah. I was good fun. Thanks for having me.
Thank you.
Oh, we love you. You're
Thank
terrific.
you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.